Good evening to all of our colleagues and patients in Europe, UK, and Ireland. Good afternoon for South America, and good morning for US, Canada, and Mexico. That should be really a busy time for you during a day. Late time for Asia and Australia. And thank you for everyone who joined our webinar today. That's a great pleasure to stay tuned with you online. Also, we are recording our webinar, so you will have an opportunity to join us anytime later. My name is Dr. Uljana Dorofeyeva, and together with my colleague Birol, Aydin, we are presenting very important series of the webinar held together by Ovogene and International Fertility Group regarding mitochondria, different treatment techniques which our team successfully practiced for years already, and the results being achieved. We are going to have a Q&A session after our webinar. So we are having 2 lectures, and after the presentations, the Q&A session. So feel free to leave your questions also as comments, and we will be happy to address them later. So during all our previous webinars, mostly we were talking about oocytes, their morphological characteristics, artificial intelligence selection and grading for the best potential results achieved, structure, and the function. Today we will go deeper into a cell and we'll talk about mitochondria as an essential, not only for the genetic disorders, but also important as an energy supply and critical factor for affecting outcome for advanced maternal age of the patients. So what we know right now regarding the mitochondria, and definitely we are keep learning day by day. However, it plays an essential role for generating energy for embryo development and maintaining embryo metabolism through key cellular functions, including the ion homeostasis, amino acid metabolism, glycolysis, fatty acid metabolism, signal transduction, and the apoptotic regulations. As well as recent literature suggests that the mitochondria content and the function may be related to implantation success and the embryo viability. That's why it's so crucial for the IVF activities and the role and the understanding of the function. It consists of the 37 genes, which encodes the RNA and the protein polypeptides, as well as we know that the numbers of the copies of the mitochondrial DNA are present in almost in every cell in our body, and that's essential for the growth and the further development. If we are talking about oocytes, so the very recent cells or the very early stages of the oocyte development, which are oogonia, consist already about 200 copies of the mitochondria, and the number constantly increases as the oocyte proceeds through the meiosis, reaches the number of about 100,000 copies up to 550,000 copies at the metaphase 2. Mature oocyte has some of the highest mitochondrial content among all the human cells, likely due to the great energy requirements of fertilization and early embryonic development. Oocytes with higher mitochondrial DNA copies numbers appear more likely to be mature, and the copies of the mitochondrial DNA numbers increases from the germinal vesicle on the stages of the GV of the oocyte to the metaphase 1 and the metaphase 2. Very important to understand that 99.9% of the mitochondrial DNA is inherited from the mother. Sperm carriers limited mitochondrial DNA around the tail, and it's about 100 mitochondria, while the egg cell is up to 100,000. Mitochondrial diseases are the mutations which can lead to the fatal inherited diseases. They can manifest at any age and in any tissue and the system. Diverse phenotypes affect almost every organ and the system, and so far there is no cure of the effective treatment except the treatment on the IVF stages. That's why the world first as the mitochondrial donation regulation came into force in 2015, and that was due to the fact of one patient who has lost 7 kids with the severe mitochondrial diseases. She sent a letter to the British Parliament asking to legalize the procedure, and she succeeded. Due to this fact, we understand that these procedures are well regulated and allowed, not really globally. Just recently in Australia, there was a new law coming into force. It was October 1, 2022. So also the regulations suggest that the mitochondrial treatment should be recommended by the professionals to the couples which really require this treatment. What about the babies which were born globally using the methods? We have publications and different groups are working. It's still a very regulated topic. However, the first world baby called as a 3-parent baby boy was born on the 6th of April 2015. The mother was a carrier of the late syndrome, as well as the first British patient who initiated the law change. And the treatment itself was done in New York. However, the transplantation of the embryos was performed in Mexico, while the first baby was born. Ukraine is the world's second 3-parent baby, was conceived for the 34-year-old woman, and the baby was born on the 5th of January 2015. That's a Ukrainian clinic called Nadia, led by the Valery Zukin. However, many more babies were born in Ukraine, also as a result of our group activities for Ukrainian couples and couples who came from all over the globe. In Greece, under the successful clinical experiments, while the mitochondrial replacement therapy was done for the 32-year-old Greek woman, she conceived as a 3-parent baby boy on the 9th of April 2019, and that was a maternal spindle transfer technique by 2 European fertility companies. And the child is labeled as a global first because that child was treated not for the mitochondrial diseases for the mother. However, that was due to the advanced maternal age and the previous unsuccessful treatments. So that was not a genetic disorder. So we also need to understand that while the ovarian reserve is decreasing with the age of the patient, as well as the mitochondrial activity is decreasing. So we really need a solution for the age-related patients. As the number of the mitochondrial DNA going down, and we can see the mitochondrial changes, increases the deletions in the mitochondrial DNA, which leads us to less production of the energy, as well as the lower IVF success rates, lower fertilization, higher aneuploidity rates for the embryos. As well as success rate depends on the oocyte quality, but not only the oocyte quality, however, also the mitochondrial activity. And we know that the results could still be really high, over 50% of the clinical pregnancy rates, in case we are using young and healthy oocyte. However, not every patient could have frozen oocytes. And if we are talking about oocyte donation as the only treatment for advanced maternal age patients, the difference in terms of the genetic relations of the baby while using regular oocyte donation, baby is receiving 50% of the genetic material from the father, and there is no genetic material from the mother. So then while we are using mitochondrial donation, 98% genetically baby is related to her parents, to mother and the father, and only up to 2% of the DNA, which is coming from the mitochondrial DNA genes, which we were talking on the beginning, 13 genes, are related to the donor. So that's why that's a really alternative for those patients who would like to try to get their genetically related baby. How the full IVF treatment cycle for the mitochondrial donation looks like. So we need to achieve oocyte from the mother. That's why we need really to address the situation in terms of the antral follicle count on the IMH level. We need to proceed with the ovarian stimulation. Of course, quite often that's not the first stimulation of the patient, and we need to analyze what was the previous outcome in order to achieve the most optimal number and the maximum number, because quite often that's a patient in their advanced maternal age, and the average age of our patients is about 42 years old. So sometimes we could recommend several stimulation cycles in order to accumulate the oocytes and then the further zygotes, depending either we are going to proceed with the spindle transfer or the pronuclear transfer technique. And once we have a reasonable number, also that decision is coming together by the clinical team as a doctor and the embryologist and the patient. We may proceed with the treatment of the pronuclear or the spindle transfer, as well as, of course, we need a donor oocytes. So donor oocytes are as well essential for the success of the mitochondrial donation, because in addition to the fact that we need to check precisely the morphology of the donor oocytes, this is what we do routinely for all of our cases. Very important is to control the fact that the donor who is going to be used for the mitochondrial donation treatment is a proven fertility donor. By proven fertility donor, we understand both being proven having her own children born and healthy, as well as the children and the clinical outcomes achieved and the children born while using the oocyte donation. As well as donors should have, and we are controlling this absolutely strictly, should have an excellent previous efficiency results, while that should be great fertilization rate, blastulation outcomes, clinical pregnancy rates, and the live births, as I mentioned, as well as for the mitochondrial donation techniques, the donor oocytes should be fresh. We, being an egg bank, we already may prove that for the regular oocyte donation, usage of the vitrified oocytes, this is something what really should be recommended because of the many benefits. First is immediate availability of the oocytes, high variety of the selection of the donor oocytes, and the absolutely equal success rates in terms of the clinical performances and the embryological performances while using fresh or frozen vitrified oocytes. However, for the mitochondrial treatments, donor oocytes should be fresh. That's why we are performing synchronization of the cycle of the donor. Donor is recommended and selected by our clinical team based on the parameters which we mentioned. We synchronize this cycle with the patient, and we are performing the treatment itself. We are going to proceed to the treatment immediately right now, and that will be presented by Birol. Just what I would like to mention that the genetic status of the embryos, we are also checking, and this is important to be addressed. In terms of the bioethics and the safety regulations globally, as soon as the technique has been allowed and proven as the one being used in the UK, the Council of the Bioethics also remarked the techniques to be safe, secured, and applicable. Some of the ethical issues are still arrived in terms of how the donors should be selected. Should the donor be anonymous or non-anonymous or known? What is the modifications and the relationships between the donor and the patient, etc. So for our practical experience, we keep all the database regarding the donors who were donors for the mitochondrial treatment for our patients. This is crucial for the results and the clinical outcomes. So now I'm going to present Birol Aydin, my dear colleague, who is a magician for the mitochondrial treatments. and he will tell more in details about the techniques and our practical experience as well.
Thank you, Dr. Uljana. Hello, everyone. So just I will start the presentation. Okay. So as Dr. Uljana talked most about the general clinical things, and I will try to go more details about the technical things for the mitochondrial transfer. So the first of all, we will try to understand about mitochondria. So the mitochondria, basically the main generates energy part of the cells. So of course, it's passing from the mother to child, which contains 37 genes, and the basic function of mitochondrial DNA, it can lead the inherited disease, which many of the people actually have, but they are not aware. While no one is going directly tested for the mitochondrial mutations. And as Dr. Uljana mentioned, there is no cure of effective treatment for mitochondrial disease. So as we are talking about mitochondria, just move my slides. Okay. So basically, why mitochondria is a taboo? So the mitochondrial DNA actually play an important role for the regulation of cellular metabolism, which is much important for apoptosis, programmed cell death, and also oxidative stress, which is very important for cell development, for further development as well. And the mitochondria are organelles that making a production of ATP energy, and ATP energy is the major source of energy into the cell. And mitochondrial DNA is the only 1% of the total DNA, which is not showing itself directly on the phenotype. Actually, which is making more attractive mitochondrial treatment or mitochondrial donation for everyone. And young age of mitochondrial DNA may increase, of course, ATP energy transportation by the calcium and between the mitochondria and nucleus. So this is how we are actually designing mitochondrial transfer technology, and why we are using young, high efficiency and tested egg donors. So basically, between the mitochondrial DNA and the nucleus, of course, there is need the production of the transported cells. So all this transported energy is doing by endoplasmic reticulum. So endoplasmic reticulum just producing calcium, and calcium just attaching to ATP energy from the mitochondria and just transferring to nucleus, and how cellular treatment and cellular development is proceeding. So the mostly, especially for advanced maternal age, when embryos starting to grow, the main problem, embryos stop the growing on day 3. Up to day 3, embryo is not reaching blastocyst. It is getting cleavage arrest or high fragmentation or not developing. That is because of the calcium transportation and energy transportation up to day 3. Because of the low mitochondrial DNA activity, it's not allowing embryo to reach the day 5 blastocyst. So as we talk about mitochondria is actually very complex molecules and organelles, which is connected with all intracellular mechanism. So the part of mitochondria, as you see on the video, is just carrying up energy to between all organelles, and especially for the main genetic material and DNA. So while this energy is not reaching the final place as a nucleus staying with the lack of energy, and the further development of the cell division is stopping. So many of the cells, apoptotic cells, is just making that there and making barriers, and these barriers is growing day by day, while up to 35 years old, advanced maternal age, also follicular development is getting decreased, and the quality of eggs, of course, drop, and lack of energy increasing, and that's why endometrial activity, also mitochondrial activity get dropped. So in this case, of course, the only way how we can help the cells to increase development, to increase energy, source of energy. And the source of energy can be only decreased by high quality of the mitochondria. As Dr. Uljana mentioned, there are different mitochondrial disease, which of course we can solve with the mitochondrial transfer. While we are changing mitochondria in the cells, that means we are solving mitochondrial disease problem. But still, as I mentioned, many of the patients doesn't aware about the mitochondrial mutation on them. So they are making many IVF treatments. Of course, once they get pregnant and they have a child, if they find out some mitochondrial disease, then they get even aware of the situation. But now, of course, carrier screening helping so much while they are able to check all mutations and they have consideration for the further treatment. But our topic here, when we start to work with the mitochondrial transfer, we just get target for the mitochondrial mutations and mitochondrial disease. And then we find out while those patients already wait a long time to get achieving baby, that's why they were already advanced maternal age. And we find out even they had very low quality of eggs and they were giving after mitochondrial transfer very good quality of the blastocyst. And then we start to think about why not we are not using this model for advanced maternal age. So that how start mitochondrial treatment can be an alternative for advanced maternal age. And advanced maternal age, the main things here, while mitochondrial DNA and the advanced maternal age cannot be active or high quality, actually here that publication shows 4977 BP deletion. So this is the main actually problem on the mitochondrial DNA in the advanced maternal age. So this is not allowing either oocytes get fertilized or developed well on the cleavage stage. Also, there are here different impacts of the meiosis, fertilization, and early preimplantation of embryo development. And of course, basically everyone knows advanced maternal age shows herself with the high aneuploid rate. So aneuploid embryo rate is much higher than younger age in the advanced maternal age. So when we get the advanced maternal age, of course, the basic things we can find recombination failure and different kind of dysfunctions and mitochondrial dysfunction, which show itself with the lower fertilization, increasing of the aneuploid of the embryos, also low quality blastocyst, and mostly cleavage arrest of the embryos. When we find out the molecular abnormalities in the oocyte, especially it is not happening just for advanced maternal age. It's also for the polycystic ovary. PCOS syndromes also shows itself with the low quality of embryo development. And mostly we are getting many of the eggs, but they are very low quality. Even number of the mature eggs which fertilize, they are not giving high quality of the blastocyst. So that's why mitochondrial treatment, it is not only for the mitochondrial disease, but in the same time can be alternative for advanced maternal age and also even for polycystic ovary syndromes. Here is the main treatment technologies for the mitochondrial transfer. When we talk about mitochondrial transfer, we show under mitochondrial transfer 3 different treatments. As a spindle, meiotic spindle transfer, pronuclear transfer, and the third one is cytoplasmic transfer. Even we can talk about the polar body transfer, which is keeping inside still a small spindle. But of course, when we talk about cytoplasmic transfer and polar body transfer, actually, according to my experience, I couldn't find any effectiveness on them. While they are really difficult because cytoplasmic transfer, you are just transferring the cytoplasm, you are transferring part of DNA, but still you are not able to detect exactly location of mitochondrial DNA. So that's why you are not getting exactly matching of the DNA in the different cell. And the polar body is the most difficult because it is a very small meiotic spindle inside. To detect this meiotic spindle and the transfer is really difficult. If you look about advanced maternal age, while mitochondrial DNA quantity is also correlated between euploid blastocyst and aneuploid blastocyst rate. So the mostly advanced maternal age showing itself with high quality aneuploid blastocyst. And if you see the blastocysts with the younger age group and older age group, while younger age group has higher mitochondrial DNA quality, of course have lower aneuploid rate. And while older age is the same as a cleavage stage of the embryos is the similar, but the number of the blastocysts is decreasing while mitochondrial DNA quality is decreasing. And when we talk about the mitochondrial replacement therapy, so there are different kind of option which you will find out when you make search on internet. It's calling as three-parent IVF, three-person IVF, three-parent fertilization, mitochondrial replacement. So you can find different categories. Everyone is calling different, but all this replacement therapy we are calling as a mitochondrial treatment therapies. The oocyte of the advanced maternal age, as we told, mostly low quality. So when we change the mitochondrial DNA and when we switch nucleus or spindle of the advanced maternal age patient to young donor eggs, actually there are different energy supplements here. While ATP energy is increasing, as you see, there are different kind of energy explosion on the oocyte, because young mitochondrial DNA is giving ability to transfer the energy into the eggs much faster than advanced maternal age. So that is how intracellular ability is increasing in the donor eggs. What about aneuploid rate in the embryos while we are doing mitochondrial transfer? It can affect the genetic development as well. So if we are using mitochondrial transfer, do we have chance to get high euploid embryos? Of course, as a direct answer, I can say mitochondrial transfer can't be improve the genetic compactation. So while the chromosomal abnormality is mostly connected with the direct DNA configuration. So while we are changing the mitochondrial DNA, which is only 1% of the total DNA, if we are waiting that we will solve some chromosomal problems, that is not right. But of course, when we change the mitochondrial DNA, embryo is developing much better. While morphokinetic activities of embryos is much higher than regular embryos, because advanced maternal age, mostly embryos are developing slowly, either giving very low quality of blastocyst, either stop embryo on the day 3. But when we change mitochondrial DNA, of course, embryo is then developing more dynamic. And during this development, many of self-correction is happening in embryo. This is also chromosomal. I cannot say that every chromosomal problems or abnormality can be repaired, but many of the aneuploidic chromosomal development as partial monosomy or mosaic embryos during this morphokinetic development can be repaired as well. So here is the main mechanism how it can be happen, because while we are using active mitochondrial DNA, that can be happen, the better apoptosis programmed cell death mechanism, and those can help for less mutation risk and high chromosomal activity, which can affect the repair mechanism, intracellular repair mechanism inside of the oocyte. As here, even for the during development, how embryos are making to itself to self-correction. So this is giving us an idea, okay, if embryo is making self-correction during the development, of course, that can be also possible when we change the mitochondrial DNA, and to increase source of energy, it can make also help for self-repair mechanism. As we talk, I say there are two main technologies, two main techniques for the mitochondrial transfer. The one of them is the meiotic spindle. So meiotic spindle, which carry up female DNA, 99% of female DNA inside. So as we say, 1% of the total DNA was mitochondria. Spindle, which we cannot directly detect by regular microscope, but which we can detect by the pulcoscopy microscope, and we can find the location of the meiotic spindle, which is important to detect the right place and to transfer spindle to young donor oocytes. And the spindle will carry up all the genetic potential into the donor eggs, as 99% of the DNA will come from the couple, and 1% of the DNA only will be mitochondrial DNA from the donor in this case. When we make the mitochondrial transfer, and as a meiotic spindle transfer, the spindle of chromosome we are removing from the donor and discarding, because we want the donor egg will stay with the mitochondrial DNA regular structure, because donor has already fertility proven donor, at least donor should give live birth, or donor should have child, or some recipient from the egg donor should have live birth. So that means that donor is avoid totally from the mitochondrial disease and have active mitochondrial DNA. So then we are taking the spindle, pick up the spindle from the couple eggs, from the mother eggs, and just we are transferring into the donor eggs. In this case, we are using the structure of the donor eggs, high quality of structure, high quality of cytoplasmic activity, and high quality of the mitochondrial DNA. Just we are transferring into the donor egg that female DNA. So then we are creating here two genes. One gene is coming from female and another gene is coming from donor. When we fertilize this donor eggs with the partner sperm, and then we are creating three genes of the embryo. So it's already zygote stage from the female DNA, male DNA, and donor DNA. Of course, here is the most important situation, how we will keep safe these eggs, because as advanced maternal age, we are talking almost we are getting maximum 2, 3, 4 eggs from the female, from the patient. And to protect from the degeneration risk of micromanipulation, these eggs is the main target of us. And another thing, still, when we move the spindle or pronucleus, doesn't matter, there will be around of the spindle of pronucleus different cytoplasmic DNA fragments, which can make a DNA contamination when we transfer in the donor eggs. That's why we should avoid all DNA which is around spindle or pronucleus. That's why we are calling that fusion. And when we do the fusion, we are just transferring right DNA into the spindle or into the pronucleus. And we already killed all DNA factors around the spindle or pronucleus. So this is the special molecules, actually, how we are protecting cell permeability during the microbial manipulation. We are not giving any degeneration risk. And from another side, we are making a fusion and avoid all DNA fragments around of spindle or pronucleus. So this is how system is working. With the special software, we are just determining place of the spindle, which you see with the light. You can just find out the location of spindle, and then we are going with laser, just open a small hole on the wall of the oocyte, which we are calling zona pellucida. And then with the special the pipette, we are just entering into the zona carefully. Of course, again, I said we are using special solution which we are calling nocodazole. This solution is just giving permeability to zona and protecting from the degeneration. We are carefully entering from the zona and just trying to take spindle out. Actually, of course, here for most of the embryologists, they can call that like it's not looking so difficult. Actually, technically, maybe not, but of course, to keep this oocyte safe is not that easy because this is still huge micromanipulation. And another side, when you make the fusion that we are calling fusion, here now we put in special solution. This is a kind of chickenpox virus, or that you can make electrical and just avoid all DNA cells around the spindle, and then we are transferring that into the donor eggs. In this case, of course, donor eggs will keep 99% of the couple DNA or female DNA. And another side, so as you see how spindle are looking, as a white is spindle. So around the spindle, as you see, still there are different DNAs which come from cytoplasm. So our aim of the fusion to kill all this cell around the spindle. Spindle have their own membrane. That's why any kind of virus or electrical activity can affect spindle and DNA inside. So this is the how system is working. And the similar here, as you see, spindle is determined here. Then we are opening zona, entering inside, and remove the spindle. And then, as you see, is a part of DNA still around the spindle is coming. So there is no chance to just go to take the spindle. So anyway, part of DNA will come. As you see, very concentrated chickenpox virus here. Just we are going and to try to avoid all DNAs around. So there are different key points, of course, here technically to how avoid the risk here. The main thing, of course, to have big experience for the determination of the spindle location and micromanipulation, and to make exactly the karyoplast and fusion. And as I talk, there are different technologies as polar body transfer, meiotic spindle transfer, and pronuclear transfer. If we are talking about which is more successful, of course, I can say meiotic spindle transfer and pronuclear transfer are more successful, as all publication and our experience are proved. The same issue for the pronuclear transfer, technically totally the same. Just what is the difference? Meiotic spindle transfer was the day 0 when we make egg pickup. Pronuclear transfer just in day 1 zygote stage. First, we are fertilizing female eggs with partner sperm, and at the same time we are using same partner sperm, we are fertilizing donor eggs. The next day in the zygote stage, they already have 2 pronucleus, and just we are removing donor pronucleus throwaway, and we are transferring female and couple pronucleus into the donor eggs. In this situation, we just create structure of the donor oocytes and zygote, and inside will have 99% of the couple DNA with 1% of donor mitochondrial DNA. And this is how pronuclear transfer technically happening. We are opening zona, just we are entering with the biopsy pipette, carefully just remove both pronucleus. I can say it looks more traumatic than meiotic spindle, while pronucleus is bigger than meiotic spindle. But again, after this traumatic process, of course, oocyte morphologically doesn't look like a perfectly. But while after the culturing several hours, oocyte became again the same morphological mode. So that is the how we are actually protecting oocyte with the different. The same things again, when you remove the pronucleus, still around will be part of DNA, which we need to avoid with the fusion. And for the fusion, as I said, there are electrical fusion or chickenpox virus. Both can help for fusion. If we are talking about our experience for the mitochondrial transfer, of course, there are many cases, but I specially choose some of them which are considering very difficult cases, which have the mostly all patients have many trials actually. For example, that case one, if we check, 30 years old patient from 38 years old from China, which patient has exactly mitochondrial disease, which we receive 6 mature eggs. We use spindle transfer technology, and we got and transfer one euploid 5AB quality blastocyst transfer, and patient already give healthy boy on February 2020. So the another cases again from China with low ovarian reserve, advanced maternal age, 41 years old, 11 IVF trials before, and never received any blastocyst, which we transfer with the spindle technology, one euploid 4AB blastocyst, and she gave also healthy boy in March 2020. And 39 years old patient from Ukraine, again with low ovarian reserve, 6 IVF trial before with the pronuclear transfer, which we transferred 2 embryos as 4A and 4BB, which patient give the birth in March 2021. So there are many cases like this, which we can consider. Of course, when we select which technology we will use, I mean, pronuclear or mitochondrial, this is mostly connected regarding egg quality. So when we do the egg pickup, we are just checking morphological quality of the female egg. If egg quality is really worse or really low, and then of course we are considering spindle transfer, while there is a risk of fertilization of such eggs, such low quality of eggs, and we don't consider this risk. But sometimes we are not able to determine location of spindle. That is the actual advantage of the mitochondrial transfer. If we don't have a chance to find location of spindle, we immediately use another way as a pronuclear transfer. But as I said, for the pronuclear transfer, we need a fertilized egg, we need a zygote. So of course, I will not talk about all cases, and then I will try to give information about the outcomes. So through the pronuclear transfer, if we check for fertilization rate, it's almost 93%, which is for advanced maternal age quite acceptable and nice result. Day 3 embryo rate almost 90%, but of course these two information is not making special mitochondrial transfer, while such good quality of lab with under good technology and experienced embryologist without pronuclear or mitochondrial transfer, also we can get good fertilization and day 3. But what will make mitochondrial transfer or treatment different, we will see in the blastocyst rate and euploid blastocyst rate. So if we check the blastocyst rate, almost 55% for advanced maternal age, which we are talking about age average around 40 years old. So of course, this is impressive result. And euploid blastocyst rate for that age also very high. While you know that for advanced maternal age above 38 years old, with the regular way, regular IVF or lab technologies, we cannot talk more than 35-40% of the blastocyst rate, almost even less. So that's why mitochondrial transfer is so helpful to get high quality of the blastocyst. And if we check the clinical pregnancy rate and ongoing pregnancy and live birth rate, we will find out amazing result. While, of course, we are not talking about 1,000 cases here, that our data is covering 67 cases, but still for the 67 cases for around 40 years old average, this is amazing result for clinical pregnancy rate and ongoing pregnancy rate, as almost live birth rate, almost 75%. That means all ongoing pregnancy, all clinical pregnancy mostly was continued pregnancy, only several of them for the different reason, mostly endometrial reason, while all embryo are tested, give abortion. So the live birth rate is very high. And if we check age by age, as you see, between oocyte and fertilization, almost similar. If we check, cleavage rate is almost very high and very low degeneration rate after pronuclear transfer or spindle transfer. That means from every 100 eggs, only 1 egg degenerate during the micromanipulation. And if we go to blastocyst rate, as we see, almost up to 45%, and euploid blastocyst rate up to 60%, which is impressive for such advanced maternal age. As a conclusion, of course, mitochondrial transfer techniques can be used for the mitochondrial disease, but in the same time, we can use that for low ovarian reserve and poor outcome which patient receive after IVF treatment. And pronuclear transfer and spindle transfer is the most effective mitochondrial transfer technologies. Mitochondrial transfer always require for good culture protocol and practice and micromanipulation. Blastocysts which we receive through mitochondrial transfer almost must be analyzed with PGTA testing. Fusion stage is the most important part of mitochondrial transfer, while we should avoid additional DNA fragments. And here, donor oocyte should be chosen very carefully, while donor should be for sure fertility proven, at least to give live birth from different recipients. And of course, also, which I mentioned, that doesn't mean mitochondrial transfer we are using for every cases. Of course, cases should select it carefully, which really we can give the patient benefit from mitochondrial transfer. We are selecting those patients. Every patient are not suitable for mitochondrial transfer. The mostly when a question come, okay, we have very good blastocyst development always, but we don't have euploid blastocyst. Does mitochondrial transfer will help us? Of course, like we cannot say those a mitochondrial transfer can solve this issue. So that's why for such situation, when we face out, we are just honestly sharing all information, and we are detecting all case very carefully with all tests, all embryo development stage, and also all problem during the embryo culturing. That's why how we are choosing which mitochondrial treatment technology is the right for the patient. Thank you very much.
Thank you, Birol. That was great. And also thanks from our colleagues as a comment in our chat. Also, excellent questions. Thank you, Professor Ash. And I will go just question by question. And everyone who is listening, if you have more comments or questions, please go ahead. So now we are starting our Q&A session. And the first question is about immunological compatibility of the mitochondria. Could it be easily transferred from anyone to any other person?
Actually, as a technically, of course, can be. But again, I said, like, we have to be sure about the quality of the mitochondrial DNA. That's why we are choosing fertility proven egg donor. If donor has a child, that means we are a healthy child. Of course, we know donor has a healthy child, or recipient give live birth and they have healthy child. That prove us like donor has high quality of the mitochondrial DNA and healthy mitochondrial DNA. In this case, of course, for us, as an embryologist, for me, the most easy part to transfer mitochondrial DNA, to transfer the pronucleus or genetic material from one egg to another egg, while if I know the mitochondrial DNA has high quality. So that can be between any eggs, I mean different eggs. But of course, the structure of morphological structure of eggs are very important. While from one eggs or one zygote, we are just removing the material as a patient female eggs. So we just remove. But another one we remove and then we put again. We are doing two micromanipulation. So that's why the morphological quality and capacity of the eggs should be very well. Other way, of course, degeneration risk very high.
Yeah, thank you. And I will comment also from the clinical side. We are considering immunological aspects of the infertility as really crucial as well. However, in the stages of the embryo development, as we know that the material which is coming from the father is not genetically related to the mother, and while we are having successful implantation is happening if there is no, if immune system of the mother who is going to implant the embryo is working perfectly. If we have any signs, and this is really considering before we are making an embryo transfer, any signs from the previous experience of the treatment of the patients or the clinical evidence of having immunological disorders, so we also include immunological treatment for those patients, and we do recommend immunological treatment as a highly recommended treatment for the successful implantation, because the implantation is a complex issue, and we cannot just lose the so important and very carefully created embryo as the embryo created after mitochondrial replacement therapy while not addressing the immunological issues. So that's why we are not selecting donor according to the immunological parameters to the patient, as we do not do this mostly for the regular oocyte donation, because this is in terms of the embryo development, and this is very early stage of the immunological compatibility of the cells. However, while we are checking the implantation potential of the patient, of course, we do check the immunological activity and the fact should we recommend of any immunological treatment or not, just for the successful clinical pregnancy and the live birth. So then there is a next question. Mitochondria varies from the different cells in the body. Is it the same mitochondria, for example, if I understand correctly, in the oocyte, then in the neurocell, in the fat cell, etc., etc.?
Yeah, every cells on the body has own mitochondrial energy and mitochondrial structure. So which we are talking the first stage of the cells. So the first stage of the cells as a compact oocytes, of course, there is one mitochondria, and which we are using. Those mitochondria is helping to create all immunological system of the oocyte, and is sending energy to all organelles and pronicles. So when organs start to develop in the body, of course, there are different mitochondrial sources. But in the oocytes, there are one mitochondrial sources as a mitochondrial DNA. That's why we are using this DNA and we are transferring nucleus to another or spindle to another donor eggs, which has high quality of the mitochondrial DNA and to supply the total development of the embryos. When embryos start to develop, of course, the number of mitochondrial DNA and number of mitochondria on the different part of the body will be different. So this is the difference how between the compact cell and development of the embryos.
Yeah, thank you. I think that's clear and the full answer. Then the next question is really interesting. Can mitochondria be frozen? If not, why not? And can or cannot we make mitochondrial banks?
This is very difficult. While the detection of the mitochondria is not possible with the regular way, only with the electron microscope. So there is no now under our hand to some technology to actually like spindle I can find and catch it. Yeah, but the mitochondria is not like this. Mitochondria is everywhere. I mean, it's not possible to detect and to take mitochondria and freeze. So of course, you can freeze an oocyte. When you are freezing oocyte, you are freezing also mitochondria. So the all donor eggs actually as a potential frozen mitochondria. But if we are talking about like when, for example, female on the young age, that's why we are saying social freezing is very important. So if you are not considering to get child in a young age, then you should freeze your eggs. That means you are freezing also your mitochondria same time. But just to take it to freeze mitochondria, I mean, maybe one day technologically will be possible, but now, no.
Yeah, I agree that probably that's a question from the future, most likely. So probably in the future, I wish we could say, yes, we can freeze, yes, we can change, yes, we can treat also different diseases which are not childhood diseases or which are not coming from the fact that this is a genetic mutations from the mitochondrial DNA, however, which is like Alzheimer or some others which are neurological diseases and which occur in the older age. So there is still very experimental methods and the way of treatment of these diseases by using young and healthy and active mitochondria. However, it's still more experimental, and I don't think we are not the professional experts for this kind of the somatic treatment and the recommendations there. However, there is also one question related to this as well. So another question, can we obtain mitochondria from ovaries extracted surgically from some patients operated for the gynecological diseases?
Yeah, actually, that is theoretically possible. I mean, of course, I never had chance to try that, but there are some research about that. But finally, you know, those all medical research, let's say, without efficiency, they don't mean anything. So when we don't have live baby, live birds, or which baby has a further development. The same issue was for mitochondrial transfer. Till till child gets 6, 7 years old, still also personally I had question on my mind, like how this child will grow up. But since first mitochondrial transfer happened, and I have experience from our mitochondrial transfer, like we had a live birth, how they were developing totally healthy without any problem, and then I became sure. But still there is no enough research regarding this, and as a personally I never tried, so I cannot say like directly it will work, but theoretically, yeah, the possible, if this way is working, that way also can work. But of course, like how we can implement it that into the cell, how we can transfer, and how we can use the mostly should be connected also for with the stem cell technology to grow up this. So how we can culture this embryo then to create it is like kind of totally create a new embryos. Again, we are going to totally beginning to stem cell technologies to grow up embryo or uterus or to grow up sperm from zero. Is the same issue like to use the mitochondria from different parts.
Yeah, thank you, Birol. And for now, that will be the last question. Of course, if you have more questions, dear attendants, we are happy to receive them and answer. But the last question for now is, in which cases we can use mitochondria and in which cases we cannot? I think that's related to the treatment, like in which cases we would recommend the mitochondrial treatment, in which will not.
So the cases actually who has up to 3-4 IVF treatments, either they never receive blastocyst, either they receive very low quality of blastocyst. I am talking about CC, BC, not better than this, and never happen any pregnancy. So for them, of course, mitochondrial transfer is an alternative. So polycystic ovary syndrome, which also never receive blastocyst. So total fertilization failure, actually we had several cases because total fertilization failure still big question in IVF. You can receive mature eggs, but many IVF trials, either with classic IVF, either with ICSI or conventional, they never give fertilize oocytes. But we saw with the spindle transfer several cases which we receive fertilization. So for those cases also can be an alternative. Of course, for mitochondrial disease, definitely is the main alternative we can use. And the mostly which embryos are developing as a blastocyst stage, as I said, it is coming aneuploid. The many times same aneuploid configuration, but for those cases, again, I'm saying mitochondrial transfer, it is not an alternative. So for those cases, unfortunately, only the way to go egg donation cycle. But which I am talking, if it is repeating 2-3 times with same chromosomal aneuploidy in the same cycles, in different cycles. So for those cases, is not alternative. But yeah, for the several cases, especially which get high mosaic rate or monosomic embryos, as I have experienced, it can be helpful.
Yeah, that's clear. Thank you so much. And for now, that's all questions for today. As I mentioned on the beginning, so our webinar is recorded, so you can get back into any minute of this great conversation. And thank you very much for wonderful questions. They were really deep and very important to address. Thank you everyone who joined, and that's our pleasure to stay online and turn together with you. We will get back and we will be happy. You will follow our results and we will be happy to provide more of those together for the best outcomes of our patients, also partner clinics globally. So thanks again for joining. Thank you, Birol. Thank you, our marketing team, for organizing this. And we'll stay in touch and see you again soon.